Let's move on to the last talk for this session. I'm going to introduce Benjamin Pedigo. He's another grad student from Josh Wagenstein's lab, and he's going to introduce this cool statistical model for nanoscale connectomes. Okay. Can I see my screen? Yeah, I don't. Can you? We can't hear you. You mute yourself.
Sorry, of course.
Oh, okay, cool.
Cool, you got it?
Yeah.
Great, well, thank you for that introduction. I'm excited to talk about my work on some statistical tools, kind of in the same vein as what Vivek and Ali have been talking about, and their application to nanoscale connectomics, and in particular on this Drosophila larva brain, which is the figure that you see to the right. So generally, I'm interested in using statistical techniques to help us understand connectome data. And when I say a connectome, I generally just mean some map of neural wiring, which we often treat as a network or graph. Connectomes happen to be getting bigger and bigger, and there's also just more and more of them coming out. And so I see analysis as a growing bottleneck for helping us understand these data. So today I'll just talk about a few examples of the kind of thing that I've worked on, including finding connectivity-based cell types given a new connectome, as well as testing for what I'll call homotypic affinity. So jumping into finding connectivity-based cell types, our approach at a high level is just an elaboration on spectral clustering that sort of accommodates for some of the features that are common in connectomes. So for example, in this case, we have a left and a right brain hemisphere, and we want those to be represented by this clustering process somewhat similarly. Or in our case, rather than a graph, we have a multigraph, which comes from our collaborators sort of splitting each neuron into an axon and dendrite. And that essentially just gives us a bunch of different types of edges, and we want to use that information as well. One of the nice things about using the spectral clustering based approaches is this duality with something called the stochastic block model, which is just a statistical model of networks that says that each node's connectivity is a function of which community it lives in. And so spectral clustering is a way of helping us recover some of the parameters of the statistical model for the network itself. So jumping into the clustering results, what I'm showing here is the hierarchical clustering process on the left and the right hemisphere of this Drosophila larva brain. So you can think of each neuron as a little bar, and I've colored it by some known metadata that we have. So for instance, for some neurons, we might know at least that they're like a sensory neuron, or a few different cell types that have been published in the literature already, like Kenyan cells or mushroom body related cells. And so one thing you'll see is that those cells, like in the red and the orange and the yellow, for instance, some of the ones that we knew ahead of time, are being clustered together really nicely. And one of the things that we're doing right now is sort of looking into all these other clusters. You know, like a lot of these are made up of many unknown cells. The brown shades are essentially just uncategorized like output neurons, and trying to understand what makes those clusters essentially. The other nice feature of this clustering is that you get this really consistent clustering between the left and the right hemispheres. So that's another indication that it's somewhat reasonable. But relating this back to the model that I talked about, one of the cool things that we were able to do here is sort of check the model fit that we get from our clustering. So on the y-axis, and this plot on the left here, let's see if I can pull up a laser pointer. So this y-axis essentially is just measuring the goodness of fit, more or less, of this statistical model. So what you see in green is what happens if I take this clustering and use it to fit a model to the left hemisphere of the brain, and then ask how well that model predicts the connectivity on the left hemisphere of the brain, or vice versa for the right hemisphere. But what I'm showing in purple is what happens if you fit that model to one hemisphere and then ask how well does it predict the connectivity on the other side of the brain. And one cool thing that we saw here is that as you increase the sort of granularity of this hierarchical clustering process, at some point around level 6 or 7, you reach an area where you actually start to go back down. So you get worse at predicting the connectivity on the other side of the brain. And so this was essentially an indication to us that we were probably overfitting at this point, and we said, okay, we're going to stop our clustering process here. What I'm showing on the right is two adjacency matrices, one of which is the real data, one of which is a sample from this synthetic model that I'm just talking about. Normally, my PI would want me to have everyone guess which is the real data and which is our model, but in the interest of time, I'll just tell you. And so this is, again, just another indication that the clustering and model fitting that we're doing is giving us a reasonable representation of the data. But one salient feature that we noticed in the synthetic data that's missing from the synthetic model is this idea of homotypic affinity. So here I'm just showing some synthetic data just to explain what I'm talking about. So if we imagine we have this adjacency matrix sorted into the left and the right hemisphere, a homotypic connection is just this off-diagonal band corresponding to connections from neuron A on the left to neuron A on the right. So in this case, we're saying we know that each neuron sort of has a bilateral pair on the other side of the brain. And so in the general model that I just described, that feature isn't really present. But if you look at the actual data, what I'm quantifying here essentially is how much more likely are these homotypic connections relative to what one would expect by chance. So you can kind of think of this as what you would get from this statistical model that I was just describing versus what we actually see in the data. So this is just to say that this is a very salient feature of this connectome, regardless of whether you're looking at axodendritic connections or axo-axonic connections and so forth. That's not captured by the model that I just described. So to sum up really quick, I've talked about how we can use estimation and modeling techniques from machine learning or statistics to help us understand these data sets. But we can also frame really simple questions about our data as network hypothesis tests and then just test them. So that last slide was showing, yes, homotypy is a really salient feature of this data set. The thing that I think is cool is that the combination of 1 and 2 helps us update our statistical models. So if we see that homotypy is a very strong feature of this data that's not captured by a common statistical model, we can start to sort of make better and better statistical models that bake in some of these connectome specific features like this homotypic affinity. Lastly, I just wanted to plug this Python package that we've been co-developing with Microsoft Research called Graspologic. All of the stuff that I've talked about, as well as a bunch of other things that I didn't get to here, are available as open source Python code. We have a paper sort of just at a high level describing the package, as well as one that goes into a bit more detail on the math. So lastly, I just want to thank all my co-presenters and everyone that's helped with this project, in particular Michael and Albert and Marta at the University of Cambridge, who it's their data that I've been working with, so really appreciate that. And yeah, I'll just stop there and see if there are any questions.
Thank you. Thank you, Ben, for presenting the interesting work. Yeah, I guess let me kick out the Q&A session. I have some questions. Can you go back a few slides to the homotypic thing?
Yeah, absolutely.
Yeah, homotypic affinity. So my understanding of this is you also mentioned this. Just I try to reiterate your statement is you actually find a very homogeneous type of neurons across hemispheres. Like if you have neuron on the left hemisphere, maybe not necessarily hemispheres, but like if you have one set of neuron here and you have another set of neuron here, if they have very strong like cross correlation, that means they are homotypic. Is that right?
Yeah, so I guess I didn't get to talk about this in too much detail, but what it is I think I might have a slide on it. So, so basically at this scale, you can find individual neurons on the left and the right hemisphere of the brain that are thought to be homologs of each other. And you can see that based on both morphology as well as connectivity. So like if you see these, this is a pair of neurons on the left and the right side that are morphologically very similar. In terms of connectivity, they're sort of mirror images of one another. So that's what I mean when I say like neuron pairs.
Does it only rely on the functional activity, or it's more like connectome? How do you evaluate these affinity?
Yeah, so for the most part, it's, I'm working, the data set that I'm working with is an electron microscopy reconstruction. So they have the morphology of each neuron as well as the connectivity. And in terms of both of those features, they, for the most part, most neurons in the brain, you can actually find the corresponding pair on the other side. I know they also do some, calcium imaging and stuff like that, and I'm not actually sure to what extent they've quantified similarity at like an activity level. I don't know the answer to that.
I see. Okay, so it's more like morphology and structural kind of similarity or affinity across the brains.
Yeah, exactly.
A question from the audience: do you think multi-D graphs? Actually, I don't know what is multi-D graph, or could represent
directed graph.
Maybe multi-directed graph could represent better nanoscale connectomes.
Yeah, so possibly, right? There's, if you treat the connectome as a graph, it's a really nice kind of appealing representation because there's so much math and stuff around it. But, there is a lot that you're throwing out, right? So, for instance, in this project, our co-authors, like I mentioned, have started to look at connections between axons and trying to treat those separately from connections between axons and dendrites, for instance. Or you could look at in some cases, one feature that we're probably not capturing in a lot of cases is like what neurotransmitter is actually happening at these different synapses. That's information that you usually don't get from electron microscopy, but it's like a very real feature of the data. So kind of separating things into a multi-graph, I think lets you add a little bit more of those complications if you want, or if you have that data.
Okay, thanks.
Not sure if that answers the question, but
yeah, hopefully that answered the question. We probably have time for a last question. If we don't have a pop-up question from the audience, actually, I have a last question I want to ask, and also related to the one that on the screen you're showing here is the statistical modeling of this multiple connectomes. Yeah. Maybe that's what you want to kind of talk about. So you said this kind of model can be used to quantify the stereotype structure versus variable structures, probably across brains or across animals. And this kind of reminds me the big hierarchical trees that you show earlier, and also the results you show later, that you have a peak actually at some hierarchy, and when you go beyond that, the performance drops. Does it imply that even across hemispheres, you are able to find some level of consistency across hemisphere, but when you go beyond that, you will run into this diverse structures or different structures across two brains that lead to the overfitting issue?
It's a wonderful question. So, in a lot of cases, generating like a connectome at this scale is already so time consuming that we often have like one brain or half a brain. And so even just getting to look at how much symmetry is there between these two hemispheres of the brain is something that, we're pursuing with this project. And like you mentioned, we looked at a little bit with that stochastic block modeling stuff. But yeah, I think it would be really interesting to use models like this to actually quantify differences. Like even if you just had two connectomes from like an isogenic, two flies essentially, and being able to actually quantify how much is preserved. Do you have preservation in sort of this high level like stochastic block model structure versus preservation at the level of single synapses? I think that's a question that, for the most part, we haven't been able to answer, but I know people are starting to collect like multiple connectomes from the same organism. So there was just a paper that did that for C. elegans and looked at it, how it changes over development. So I think that's a really exciting area of research.
Thanks. Yeah, that makes me exciting too. Listening to all these possible directions, problems to be solved in the area. Okay, yeah, I think that's basically the end of this session. And let's thank again all the speakers, Vivek,